CLL cells proliferate in the lymph node tissues, and they proliferate not in isolation, but in contact with what we call the microenvironment, which are other cells, which in the end activate signaling in the leukemia cells, which is critical for the proliferation of the cells. Going on to say, well, if we follow that concept, the question becomes which pathways are important and critical, and with that, what we've learned over the last years is that the B-cell receptor signaling pathway is a critical pathway, which we now know from laboratory studies, but we also know that from the clinic, where we use the new targeted drugs that are blocking B-cell receptor signaling. And the key molecules are the BTK inhibitors. There are two approved now, which are Ibrutinib and Acalabrutinib. They are blocking B-cell receptor signaling, and they are highly successful in the clinic, and they have become, over the last years, probably the most effective and widely used standard treatments for patients with chronic lymphocytic leukemia. So that is kind of the introduction. From there, I'm talking about two specific projects, highlighting things we've done in terms of correlative studies, how to measure and to quantify the effect of new treatments on proliferation of CLL cells. We've done a clinical study a few years back, and I'm just going over these data again, where CLL patients were drinking deuterated water to label the CLL cells in a way that you can quantify the proliferation of CLL leukemia cells in the patients, and then you can quantify how that's affected by Ibrutinib treatment. We've done that in this study, and I'm discussing the results. The key results are that once you start treatment with these B-cell receptor signaling inhibitors, the proliferation of the leukemia cells is effectively shut off, and then the cells start dying. So I'm bringing that as a correlative clinical example, how these new treatments are not really cytotoxic treatments, but they are indirectly shutting off the signals that make leukemia cells in CLL proliferate. And then I'm highlighting another study which we did in collaboration with a group in Uruguay, a very interesting study about activation-induced deaminase, which is basically an enzyme which in normal B cells induces point mutations in the B-cell receptor to help normal B cells with what is called affinity maturation and isotype switching, where these point mutations help to build the best possible antibodies, which then have high affinity to the target antigen. So in CLL, there was a study maybe 3 or 4 years back suggesting that treatment with Ibrutinib, Idelalisib, with this class of agents that inhibit B-cell receptor signaling, that these agents would upregulate the expression of this enzyme AID. And consequently, there was concern that when you use these medications, which are now our standard treatment, that you would induce over time maybe mutations which could lead to either drug resistance or maybe to other cancers. So there was, based on that, quite a bit of concern. But those studies were largely based on cell line data in a cell line which is called MEC1. And we did a study to follow up on these results using primary CLL cells from patients treated with Ibrutinib, and we looked at proliferation markers in these leukemia cells, and we looked at AID expression, and we saw that the AID expression in those treated patients is actually coming down along with the signature of cell proliferation in CLL cells. And we stimulated primary CLL cells to express AID by using stimulation through CD40 ligand and interleukin-4 stimulation, and you can in that way induce AID expression. But when you do that together with these kinase inhibitors, with Ibrutinib, then you down-modulate AID expression, you don't upregulate it. When we took the MEK1 cells and treated them with the agents, we saw what has been published in the cell lines. This enzyme becomes upregulated. So the mechanism, cell lines versus primary leukemia cells, seem to be quite different, but our impression from our results is that AID goes down and the proliferation of CLL cells is down-modulated by these treatments. And we feel, and that's now the experience using these agents for about 10 years, up to 10 years, is that these agents are safe and are not inducing these mutagenic events in CLL patients. So then I'm summarizing to say the proliferation of the CLL cells is happening in the lymph nodes. It is to a large extent driven through the BCR pathway, and then we're fortunate to have now these new exciting drugs which can interfere with these signaling pathways, and which are a good treatment now, have become a very good alternative to what we used in the past, which was chemotherapy-based treatment.